by Professor Alexey Zaikin. He will tell a bit more about himself and in general. So please welcome our speaker.
Right, okay. So my name is Alexey Zaikin. I'm from University College London, where I have a very interesting, but now probably common position. It's a chair, so professorship on computational medicine between Department of Mathematics and Institute for Women's Health. In Institute for Women's Health, I communicate with real clinicians, whereas in Department of Mathematics, with mathematicians. And correspondingly, despite the fact that actually my research direction is medical cybernetics, so I'm mostly interested how complex physiological systems process information. Two sub-directions include analysis of clinical data and modeling of physiological systems displaying some kind of intelligence. I forgot, first of all, no, it's not first of all, let me thank sponsors of our conference as well as organizers, so thank you very much, and Professor Zaikin also, who has recommended me to this conference. Hopefully he will not regret after this talk. I will speak today about Synolitic AI, and of course I will explain you what does it mean. Synolitic, and I have put here also a logo of my group, so you can see it here a little bit larger, together with the secret title of my talk. So this logo actually is a very famous Vitruvian man of Leonardo da Vinci, redrawn as Vitruvian woman, because I work at the Institute for Women's Health. And mostly interesting, in the description of this picture, Leonardo was writing that the most fascinating fact is that the laws that describe our universe are the same. The laws are describing our human body. And this is a typical approach of system science, including system medicine and systems biology. We are interested to detect dependencies which will be system independent, in other words, quite general. And today I will speak about Synolitic networks, or in particular, how to construct network biomarkers if there is no network. So here, I will start with finances. It probably is the most important thing. You can see what happened with financial market. You know, of course, on 27 January 2025, after release of DeepSeek R1. And as the loss was $1 trillion per day in comparison to probably $29 billion for ChatGPT and just 500 billions invested exactly the same day after Trump announced this investment. And DeepSeek itself was costing just $6 million. But why I'm showing this picture is, as far as I know, and I'm saying as far as I know because in front of so many young people able to program much better than I would be able even to program till the end of my life, I don't want to introduce myself as the best expert in AI. I will speak mostly about challenges in analysis of clinical data. And that's why it will be highly important that you will provide me with some feedback and tell me your new ideas, because I'm very much open to cooperation. But as far as I know, DeepSeek has used the following idea: a layer or ensemble of low-dimensional classifiers, then have voted, and that is why both the training and voting was more fast and more effective, and that's why the cost was just $6 million. And this is actually the same idea that I'm utilizing in my Synolitic networks, this ensemble of low-dimensional classifiers. And you can see that such ideas can be quite productive. One can lose up to $1 trillion just in one day. But now, step by step, we will speak about analysis of clinical data. So the question is, how many numbers actually do we need to characterize the human body? Well, first of all, we have, of course, a human genome. And by the way, sequencing of human genome has been finished just 2023, just 2 years ago, when 179 additional genes have been found, and it started, of course, 25 years ago. So here the cost was approximately $3 billion for human genome. Now you can probably with 23andMe or other companies sequence it for $70. And human genome, we have 2.3 gigabyte of data, approximately the same amount as in my iPhone. I'm afraid I forgot my iPhone in the toilet. Yeah. I'll stop for a second. Yeah. So this is approximately 2.3 billions of data. Maybe this is not so much, but the problem is that, and you can think that in all our cells, genome is the same. But actually, it's not always the same. Just one example, under the stress, neurons in our brain can break all strands of the DNA, and then genome will be different. Another important point is that all these genomes in different cells, and we have 30 trillion cells, is transcribed in a very different way. Just to give you the example, if you unfold DNA just from one cell, you will get approximately the length of 2 meters. If you couple together all DNAs from your body, you will get approximately 200 millions of kilometers. It will be 70 trips to the sun and back. And all this information is used transcribed in a very different way. Because in each genome, only 3% is responsible for protein encoding genes, and the rest 97% is responsible to read information from this 3% of the DNA. So it's extremely, we are an extremely complicated set of numbers used in unknown way. Then, according to the genome, we have a chromatin assembly, because reading from the DNA depends how it is folded or unfolded. Additionally, we have methylome, so we played a lot with analysis of DNA methylation data. It's simply an original signal which can be added chemically on the region CPG of the DNA between C and G. And if, for example, inside the gene you have a lot of methylation in the beginning of the gene, in the promoter, then this gene will be silenced. For example, if you smoke, or during the aging, or during the cancer, some genes will be silenced, and one can get some problems. At the moment, so I was starting maybe 15 years ago, analyze methylation data when the resolution was 26,000 of probes per genome. Now we can measure it with 1 million of probes per genome. And this will be different in different cells. Because we have 30 trillion cells, and additionally we have 30 trillions of other cells living in and outside of our body, organizing a microbiome. In terms of the mass, this would be approximately 1 or 2 kilograms. So right now, 1 or 2 kilograms of other creatures are living in or on our body. And this microbiome is also important. Just to give you 2 examples, centenarians, so people who manage to live after 100 years, they have a different microbiome. Or this microbiome in the gut can be responsible for the Parkinson's disease, because bacteria manage to send some information through the nerves towards our brain, and because of this gut-brain axis, we can get a Parkinson's disease. So just imagine, and now additionally we have proteome, a lot of proteins, because from this DNA proteins can be expressed, and they are expressed not directly, the gene protein, they are expressed through the splicing, so different regions of the gene can be spliced in a different way, organizing from 20,000 genes more than 1 million proteins. Fortunately, we can measure it, measuring proteome with mass spectrometry. So just imagine how many numbers we have now to characterize our state. But what is important is that this huge number of numbers is not static. And additionally, we have, of course, images like MRI, fMRI, computer tomography, ultrasound, or histopathological microscopic images. And of course, you know that any image can be represented in the form of the very high-dimensional vector. But what's important is that these numbers are linked in the networks. Inside each cell, for example, on the level of the genome, we have transcriptomic regulation, so all genes are organized gene-regulating network. And all these networks have some intellectual abilities. For example, I started with DNA. Just on the basis of polymerase reaction, one can recognize a Hopfield neural network. Then we have genes, so gene transcriptomics. I have some papers to organize associative perceptron. You need just 3 genes, and this system will then work like the associative perceptron, working like the Pavlov dog, associating, for example, 2 stimuli. To organize the summating perceptron, you need just 5 genes, and we have 20,000 in each cell. So the whole network is very, it's in fact artificial intelligence device. An amoeba, unicellular organisms can actually demonstrate this ability, in particular solving NP-hard problems in linear time, so much quicker than it will be required by a simple enumeration of solutions. Cells are organized in organs, and organs are also intelligent. For example, there are works to show that kidney is a perfect artificial intelligence device. I'm not yet speaking about our brain. Our immune system is an intelligent system. Finally, on the top of this, we have our brain, where we have 86 billions of neurons with 10,000 connections each, coupled in very complicated network. On the top of it, we have local integrators, glial cells, astrocytes, again, approximately the same number, 86 billions of cells. So all this huge set of numbers to characterize state is actually linked, organized in a kind of a super network. But that's not all. This super network is evolving in time, because we can have a disease, we are living, we actually can transmit a lot of high-frequency signals, like bracing, muscle tonus, EEG, ECG, and a lot of fMRI, which is also time series image we can move. So all this super network evolves. And then unfortunately we have aging or a disease. So now I hope it's clear that in order to characterize the system, we need network biomarkers. Why? Because we need to classify disease and sometimes, of course, first of all, to get a diagnosis. Or, for example, how in the case of glioblastoma, if you take a biopsy, you should predict which chemotherapy should be used by artificial intelligence, because it should be much faster than developing this chemotherapy by usual biological experiments. It will be simply not enough. The patient will die. Now, we need early detection, like, for example, in the case of ovarian cancer, where if you wait just half a year, mortality, 5 years mortality changes, I will show these numbers, just from 90% to 19%. And we need it for active survivors, like, for example, in the case of prostate cancer, where sometimes treatment or even only taking a biopsy can be more dangerous than not treating this cancer if it's not a progressive case at all. So we need network biomarkers. Unfortunately, now we have, so the situation is that we have more information that we can analyze, but fortunately, of course, we have now AI, so convolutional neural networks, recurrent neural networks, LSTM and GRU, GANs, and of course, transformers, with ChatGPT and with LLMs. And of course, we use this artificial ready schemes a lot. Just to give you some example, in data-driven analysis, we use it for analysis of prostate cancer progression. We use ensemble learning for pancreatic, for detection, early detection of pancreatic cancer, or we use LSTM methods for ovarian cancer. We use also these ideas of artificial intelligence for biological intelligence, especially to show the role of astrocytes in organization of situation-based neuromorphic memory and associative learning. We use this principle to understand how we can organize artificial intelligence in engineered bacteria. This is ideas behind synthetic biology, but the situation is still very bad, and very bad because of course, because of actually 3 points. First is the course of dimensionality. So dimensionality of medical data is usually much higher than the number of available patients, and as a result, in the complicated space of parameters, you can finish in the local minima of optimization instead of the global one. Second point, it's very difficult to detect hidden topological changes because we are a super network, and changes can occur in very different places. And the third point is the interpretability. Very usually, a neural network is just a black box, and it's not clear how to interpret these results. And that is why it's not clear how to find the target for medical treatment. That is why I observe now 2 very important directions that happen with AI. One is link with actually fundamental mathematics. We have heard today fantastic talk of Barannikov about TDA. So especially with graph theory, where we can observe appearance of deep geometric learning, including graph neural networks and of course knowledge graphs. And another direction is the link between AI and nonlinear dynamics, what I like very much because I have spent actually 15 years of my first research in nonlinear dynamics, where we have spiking neural networks, reservoir computers, which work much more effective for the prediction of time series than RNNs or LSTMs. I don't know how good they are working in comparison of the transformer, but now people in fact are very interested about reservoir transformers and about graph reservoir transformers. And also here we can speak, so we are very much interested in such dynamic effect as, for example, noise-induced artificial intelligence. Maybe I will show you a couple of slides, but if not, you can simply Google. We have shown that stochasticity or complete randomness in fact can make system intelligent and operating as artificial intelligence. We are also very much interested in consciousness, and for example, recently we have got a dependence between consciousness and intelligence, and surprisingly this was non-monotonous. So to get the best, the high level of consciousness, you should not have the best intelligence. That's why probably computer is, I think, more intelligent than me, but I hope less conscious. Anyway, I will speak today about this link between AI and graph theory, and of course about analysis of systems represented in the form of the network, but especially I will speak about synolytic networks, or how to represent the system in the form of the network if there is no connections unknown a priori, so if there is no network. But first of all, why do we need to analyze network biomarkers? For this, let me cite a paper was in Scientific American, and they have written, and it's actually now well-established fact, that one difficulty, it was about actually commenting on cancer genome atlas. And they have written that one difficulty in interpreting this data for defining clinically useful information is that multiple different changes may be responsible for the onset of a disease. Why can this happen? Well, suppose I have just discussed that our human body is a super network, and it's clear if a lot of features are connected, and we have, for example, a problem here, our nature of our human body has a lot of mechanisms to compensate this malfunction. And if the problem was here, probably it will be compensated, and then biomarker linked with this node will not show any changes, but because of this connection, something will happen either here, or so somewhere. So the topology of the network will be changed, but some concrete biomarkers will not be changed. And in fact, it's a very common case. Just a very good example, ovarian cancer. For this, we have a biomarker, CA125, and it perfectly displays increase for 85% of cases, but for 15% of cases, there is no increase in this marker. My hypothesis is that this happened because of this compensations. And of course, if we know the underlying network, we should analyze it. Just for illustration, here is the genetic landscape of the cell. But what to do if there is no network? So if how to construct a network if links are unknown? Well, several approaches have been used. So here again, it's an illustration what I'm going to speak about. So suppose that you have parameters describing human body, state of your human body, but links between them are unknown. How to construct a network? Well, for example, one can use correlation graphs. And recently there was, it was 2021, it was a very good review by Alexander Gorban about dynamic and thermodynamic models of adaptation. We have also contributed to this review about our work about paralytic and synolytic graphs. And actually the idea is if you have time series, so multiple channels of time series, for example, EEG, then you can compute correlations between them, and each channel will be the node, and the weight of the link will be the corresponding correlation. So then each patient or population, for example, can be represented in the form of the correlation graph. There is a very strong fundamental principle behind changes in the correlation graph, because from thermodynamic principles, it's known that before a catastrophe, and disease is actually a catastrophe for the human body, or mathematically speaking, we can say it's a bifurcation, which can be with slow change of the order parameter, or probably with a jump, so very abrupt change of the order parameter. So before the catastrophe, we have increase in the correlation and increase in the variance. After the catastrophe, so after the onset of the disease, if it's a case of disadaptation, so the system didn't manage to adapt to this, the correlations are lost, but variance is still very large. So one can use correlation graphs, but this can be used if you have time series. What to do if you don't have time series? You have just the parameters. Probably, and by the way, I forgot the slide, this idea can be used, of course, in analysis of fMRI data with graph neural networks, because the usual pipeline looks like following. From fMRI, we map it on ROI, regions of interest, this parcellation atlas of different regions in the human brain is available. Then we identify a signal, so averaging over this area, signal corresponding to each area, and construct correlations between them. So we construct a connectivity matrix, which of course each matrix can be represented as a graph. We use graph representation, and then we can use, for example, convolutional graph neural network to characterize the brain state. But again, this is possible if we have time series, because fMRI actually is MRI in time. What to do if we don't have time series? Probably we have some internal profile of each parameter. Like in the case of methylation for each gene. So here we have just a methylation signal for one gene, this BRCA TNF something. BRCA genes are actually can be linked with the breast cancer. You know that Angelina Jolie needed to cut her breast because BRCA genes were corresponding to the very high probability of breast cancer. Anyway, we can construct a methylation profile for one gene, because this was done still with the resolution 450 probes per gene, so approximately 17 points per one gene. And you can see, first of all, the signal inside each parameter. You can construct this profile if, for example, you use some averaging window. And for example, you can see that in healthy patients it will be like the blue line, so more or less the same, whereas in cancer patients you will get some stochasticity. It will be very much spread about this profile, which can be also identified with this color map. So in healthy patients, more or less profile is the same, whereas in cancer we are getting a very large spread. So if we have internal structure of the parameter, you can of course compute correlation between these parameters. In this case, one should use a canonical correlation applied if lengths of vectors are different. And if you compute, that's what we have done in this paper about intragene. First paper was to show that intragene DNA methylation architecture is important. The second was to show how to identify network encomarkers, because we can look for the associations between this correlation and, for example, rate of survival, so ability to survive. And then we can denote it by green if correlation is positive, and by red if correlation is negative, and took connected structures, and in this way identify a large network biomarkers of survivals based on the analysis of DNA methylation. And if additionally we will do gene enrichment analysis, so we will check which genes are responsible for which function and look, for example, only for genes responsible for immune system, we can identify small network biomarker, for example, responsible for the immune model or for the healing model, and so on. But this is possible if we have internal structure inside each parameter, because one can compute the correlation. What to do if you do not have any information? So simply you measure proteomic structure of your blood, and you are getting 300 concentrations of different proteins. So currently I'm, for example, characterized with 300 numbers, and links between them are unknown. Some breakthrough happened when Stefano Bacaletti and Massimiliano Zanin in 2014 suggested the idea of parenclitic networks. Parenclitic, from Greek word parenklisis, means deviation from normality. Idea was very simple. We will put all, for example, proteins like the nodes, and we will reconstruct the H weight, how much these two parameters deviate from the normality. How this can be done? For example, for these two parameters, these two proteins, we plot the points corresponding to healthy patients, for example, controls, green triangles, then reconstruct a linear regression between them, we put the linear regression, and then for each new patient, for example, some red point, we measure the distance from this concrete point to this regression line. Of course, before we need to do some normalization, for example, to measure in standard deviation. So that's correct, typical normalization procedure. And this distance from this linear regression will be the weight of this H. Then each patient will be represented in the form of the graph, where weights are actually deviations from the normality. And this is the idea of a parenclitic graph. Idea is actually quite interesting, and we use it for analysis of cancer identification using methylation data from Cancer Genome Atlas, and you see that even visually, so this is the case of cancer, and this is the case of the healthy patients. So here we are getting much more connected structure with some star-like nodes, so nodes with very high number of degrees. But we were not so much happy with this because sometimes it can happen that cancer patients or cases will be simply in this, in the plane for these two parameters, will be shifted along the regression line, like I have illustrated it here. So more or less these red points have the same directions from this regression line as these green points, but they are simply shifted. So what we have suggested KDE technique, kernel density estimation. In other words, simply for each new point, doesn't matter in which direction, to measure the distance from this point and the cloud of healthy patients, and to put this distance in the H weight. Now it doesn't matter in which direction cloud of unhealthy patients will be shifted, we will get the same weight. And we have applied for the predicting ovarian cancer, and again, even visually, you can see, so if the diagnosis is more than 40 months between ovarian cancer, structure will be different if the diagnosis, less than 14 months, if the diagnosis is larger than 36 months. So, in fact, more than 3 years before the ovarian cancer. And this was from the screening program. I will speak about this a little bit later. But again, we were not so much happy because we didn't use cases, so unhealthy patients in the contraction of the model. And then it was this COVID-19 pandemic, so this scary 2020, and we are proud to say that already in the December 2020, Guardian has written about our large study, which started in March 2020, so patients admitted to the hospital Charité in Berlin have been analyzed, measured, and then in December we started to publish our results, and PI of this study, a doctor from Charité Clinic, said that we can predict which patients will need oxygen support and ventilator quite accurately, and we also have markers for patients who are not that severely ill initially, but at high risk of getting worse, because as you remember probably in this COVID it was a very common situation that the patient initially didn't show something bad, some bad symptoms, but then suddenly died. So we have lost, unfortunately, a lot of people. So this was a huge collaborative effort, 61 authors, 28 affiliations. What we have done, we have analyzed the patients who were actually at the end classified according to the grade 7 of the classification from World Health Organization. So patients who at the end needed ventilation or additional organ support. So altogether there was 139 patients, and among them we have analyzed applied modeling for 63 patients. Among these people 20 died, and most of them were patients with a grade 7. So in that time a lot of people were trying to identify markers of these severely unpredictable COVID symptoms. So people tried a lot of plasma proteomic or metabolomic markers. So what we have done, we have analyzed the data from high throughput proteomic analysis. High throughput means, because usually it's not high throughput, mass spectrometry, it's a huge equipment and takes a lot of time. Here it was just taken measure 180 patients per day. So it's high throughput proteomics, and 309 proteins have been measured with this scanning SWAS technique, whatever. So each patient has been characterized with 309 numbers. And we have constructed our model for these patients. So World Health Organization grade 7 patients, among which we have excluded 14 because in the time of the study, in March 2020, result was still unknown, and the rest was split in cases who unfortunately died or had were in very complicated situation, and controls. So 34 patients, and we applied the following methodology that we have called actually constructing a Synolitic network. So for each two patients, for each two features, so two protein concentrations on the plane, we plotted a cloud of patients which are controls, so healthy, who survived, and patients who didn't survive. Then with any kernel, with well-established machine learning kernel, in this case the best results have been obtained with radial support vector machines. We have obtained a boundary between healthy and unhealthy patients, and then probability to belong to the class of case 2, so of case patients. So this probability have been put as the weight of the corresponding graph. So practically, and this is what we have called as the Synolitic graph, from Synola, Greek word, which means ensemble, because this graph is actually an ensemble of independent low-dimensional classifiers. So each classifier is in fact just using two features, but then all these classifiers are linked in the graph, and we can analyze topology of this big graph, which is actually an ensemble graph or Synolitic graph. What is good is that this is actually quite natural representation, because it's a network representation of these unlinked numbers. So it's quite natural for the human body, and even you are getting it's quite quick, because for each classifier you need just two features, and probably one can overcome a problem of overfitting. And it's give you some kind of interpretability, because you can visualize even by eye a structure of this graph. Or you can, of course, characterize this graph with topological indexes like number of degrees, distribution of degrees, centraliness, betweenness, so 31 Google PageRank index, so a lot of topological indices, and we use this classification. So here just some visualization of healthy and unhealthy patients, that we can compute this topological characteristic and train a classifier. And the results were quite good. AUC, so area under the curve of the ROC curve, was 81% with leave-one out cross validation for this charity cohort. But then when we used this model trained for Charité data set, and when we have applied to the additional completely novel and independent data set obtained in the Innsbruck hospital in Austria, the accuracy was 100%. So AUC was equal to 1. This, of course, a little bit suspicious. If you see AUC equal to 1, so probably it's a kind of fluctuation, because it was actually just 19 patients, and that's why it's not so, so it can be just a fluctuation. But in any case, it was quite good, because this data set was completely independent. So we have validated our classifier with independent data set. And then we started to use this Synolitic AI in very different applications, and I will explain you some. For example, we applied to construct age-related trajectories of DNA methylation. So I told you about methylome, DNA methylation markers for patients with Down syndrome. And in this study, by the way, if you have any questions about Synolitic approach, please ask now, because this is the main slide that you should understand. Yes, ask. I will hear you. Actually.
Am I right that Synolitic network is constructed by two features?
Each weight is constructed by two features. Yes, but altogether we use all pairs of features. So between these two, we construct this boundary between these two features, and it means that we construct for these two features and independent classifiers for these two features. When a new patient is analyzed, for these two features are analyzed with this classifier, and we get this way. These two features are analyzed by another classifier. So we are getting a matrix of classifiers. For healthy and unhealthy, for the training set. So of course, as usual, we split the data in the training and test set. So we construct classifiers using training set, then apply this classifier for any new set. Yeah? Any more questions? Yes? Difficult to hear, sorry. Louder. So let me split. So of course, each graph is a matrix. So in fact, we represent, after constructing of all these pair classifiers, this level of pairwise classifiers, a vector of the patient is embedded in the graph space, which is a matrix. But each number is obtained by low-dimensional classifier. Our weight is practically the distance towards the red class, towards the class of disease.
And how we extend which exactly person from healthy part of our set to connect our...
So for training set, this training set where we know who is healthy, who not healthy, we construct this boundary. Then for a new set, we know where the point, where with his two features or her two features, we know where the point will be located, here or here, and we simply analyze the distance. Yes, and this distance towards the disease will be the weight of our edge.
Yes, but which points we should connect? How we extend this?
No, we connect all of them initially, but then you can apply some threshold, for example.
Oh, okay.
Yeah? Okay, good. Yes?
So you mean that you build a classifier for each possible pair of these nodes?
Yes.
Then you train a classifier for each of this pair of
Yes, first we train
features?
a layer of classifiers. So for each two features, we train its personal classifier.
And then for the person
I analyze all pairs separately and get all these connections separately. So the new person will be represented as a graph.
and after that you choose some threshold and
You can choose some threshold. You can or you cannot. For example, here for visualization purposes, we apply some threshold because not all connected. But in fact, for graph neural network, we apply graph neural network later, you don't need to use a threshold. So you can leave fully connected graph with different weights. It's in fact a good question to apply a threshold or not. It's one of the questions, for example, in this research project. Yeah? Okay.
We have a question in chat. So in the vertex is a classifier, and what is the edge? Is the distance between vertices equal?
Yes, so vertex is a parameter, edge is a label assigned with the class. So how much these two parameters characterize this patient as being unhealthy. Okay? Okay, let's go on. And then we started to apply the Synolitic principle, in particular for identification of molecular signature for the Down syndrome. For the Down syndrome, it's not a problem to detect the disease, but it's completely unclear what happens with underlying molecular interactions. So here we have studied 29 families where was a healthy mother, unhealthy sibling with Down syndrome, and a healthy sibling. And what we have done, we have excluded features which are clearly identifying Down syndrome and played with the rest. And what we have done, we have used some parameters of the graph versus age of the patient. So green are mothers, blue are healthy sibling, and red is a child with a Down syndrome. And then we have detected a lot of very different effects. For example, one can exactly see which feature of the graph will be distinctive from healthy people. So here it's completely different for the Down, for the child with Down syndrome, but it's the same for the healthy child and the same for the mother. Or, for example, you can see which features are responsible for the accelerated aging, because Down syndrome is actually can be interpreted as accelerated aging, and you can see that children with Down syndrome are aging towards the mothers much faster than the healthy siblings. And in fact, it was quite surprising that applying the Synolitic representation, we managed to get this trajectories. So one could see really these trajectories of the development. Or, for example, one could even see hyper-aging in people with Down syndrome. For example, here you see that red is closer to mothers than blue. Or, for example, divergence of aging trajectories. You see here for some parameters, healthy sibling is developing with age, so from 20 to 40 towards the healthy mothers, whereas Downs, for example, not. Or, for example, one could analyze a so-called biological clock. So it's very important to measure the biological age of the organism. For example, some people with age 40 can have biological age 50. Some can have biological age 30. And we have shown that this approach displays actually more precise results because for the difference, positive difference between biological age and the passport age, downs were closer to mothers, and even if the distance, if the difference were negative, again, downs were closer to mothers. So in both cases, we were able to compute this age more precisely in the Horvath's methylation clock. One possible, and what is important that for all these processes that I have enumerated, it was possible to see the exact Synolitic graph, so exact topological feature of the underlying network responsible for this behavior. And then, of course, we were interested actually how good is this Synolitic classification. So we decided to play with synthetic data, and we tried to use the data which are not so good classified by traditional machine learning algorithms or neural networks, and played with the different overlapping spheres. So, for example, I will not go now into the detail. For example, the healthy were here, and unhealthy people were in the sphere of larger radius, where some features were random, some features were simply increased, so that no traditional machine learning algorithm was able to classify such overlapped features, because in all projections you are getting an overlap between healthy and unhealthy people. And then we have, of course, visualized the Synolitic graphs. Most interestingly, what we have done, we have played with the ratio between the sample size and sphere dimension. In other words, we have played with the ratio between the number of data points and dimensionality of the space we are working in. And if you go from right to left, because here what is displayed is the logarithm of this ratio. So after 0, we are getting that dimensionality is larger than number, I call it patients, but actually some data points. So from the 0 to the left, dimensionality of the space, number of features, is larger than number of data points, number of patients. And we actually compared three techniques to construct Synolitic or paranclitic graph, because Synolitic graph is in fact a subclass of paranclitic graphs, where you use in training also training on cases. And considered a difference between AUG constructed with one of the technique of the Synolitic graph and AUG constructed with the best available machine learning techniques. And we have checked support vector machines, different XGB trees and decision trees, and of course neural networks, just fully connected neural network. And you see that when we are getting this course of dimensionality, actually Synolitic works start to work better. Doesn't matter, in fact, which technique you are using, linear regression or kernel density estimation or the proper actually Synolitic graph. And this looks very promising, and one can in fact explain it by the fact that we are using actually low dimensional classifiers to construct each edge, and that is why probably one can overcome somehow a problem of overfitting, because every time training occurs in the low dimensional space. And then of course we are coupling this with graph neural network because initially we played of course with topological indexes, but one is getting combinatorial explosion of possible best models very quickly, because one can construct 31 standard topological index. But if you use, instead of graphs, if you consider distributions of degrees and so on, you are getting, as I said, combinatorial explosion of possible estimations. And of course, one can use graph neural networks to characterize these unconnected parameters. And then what is interesting, we have got the connection between analysis, result of the analysis of the graph neural network applied to unconnected high-dimensional points. So in fact, now with this approach, you can apply graph neural network to any high-dimensional data with unknown links, because first you can construct a Synolitic graph and then apply a graph neural network. We have used basically in this study convolutional graph neural network. Here, just if you are interested in architecture shown with different convolutional layers, which with batch normalization and dropout, which is used to avoid overfitting, and then pooling layers, where you transfer classification of each node to the classification of the whole graph, and actually in all situations playing with this very different synthetic data of very complicated structure. By green it's shown when AUK was better again than any other best available machine learning or neural network algorithm. So a kind of intermediate summary. Now we are very much interested in building this Synolitic AI. Again, what happens if we have numerical data, or by the way, this can be also applied to imaging. I will speak about this because Daniil from Institute for Cognitive Neuroscience had developed quite nice representation of fMRI in embedding of fMRI image into the Synolitic graph using again pairs of voxels and how much they are changing towards another state of the human brain. They can be applied to high frequency signals because of course first we can use an encoder towards some high dimensional space, and then they apply Synolitic graph in this high dimensional space. Or now I'm very much interested in the application of graphs to electronic health records, but this is not directly. So at the moment I see that if a patient is characterized by numeric data and electronic health records, first can be embedded in the Synolitic graph, and second into the knowledge graph, and one can use graph neural network to analyze them simultaneously. So then we construct a graph, probably after thresholding, not all nodes will be connected, and analyze this graph with graph neural network. What are the advantages of this technique? That this is a very natural representation of human body, because we are a network, as I have explained at the beginning. There is hope that one will be able to overcome a problem of overfitting, of course, of dimensionality, because actually in this layer of this neural network technique, in some kind, this is just some special architecture of a neural network. We use a layer of low dimensional classifiers. So every time just two connections are used, or maybe if you want to construct a hypergraph, it's another, by the way, interesting question, can be also used in the project, then 3 connections will be here, or maybe 4, maybe 5, but in any case, the dimensionality of this classifier is much lower than original space. That's why probably no overfitting. It's quite universal, because here one can substitute any possible kernel, support vector machine, logistic regression, any kind of the XGB tree, some small neural network, whatever you want. And we are getting some interpretability, because you can visualize the result. So I have still 20 minutes now about challenges and work in progress, because now we are continuing to use this technique. So first problem is analysis of proteomic longitudinal analysis. We have access to quite unique data from UCL study. It's United Kingdom collaborative trial of ovarian cancer screening. And the more best advantage of this screening is that women were screened before onset of cancer. So after the menopausal age, women, namely 500 postmenopausal women, were screened annually until some of them have got cancer and some of them not. And all these blood samples are stored in the fridge, special fridge, and one can have access to them and analyze this blood. But you can analyze the blood 5 years before the onset of cancer, which makes, of course, this data absolutely unique. And then there were 300 80 MMS screens. Point is that there were different arms in this multimodal screening program. Some of them were control group, 100,000 women, 500 were tested, biomarker has been tested, and 50,000 were this measurement of the biomarker plus ultrasound. And now we have the development of this biomarker for the women who have got cancer before this cancer, and additionally to this ovarian cancer biomarker in this study, my postdoc, who is now associate professor at Primary University, has got this project. We measured new biomarkers, approximately more than 300 oncomarkers for lung, pancreatic, and colorectal cancer. What is important that we have these biomarkers before onset of cancer, and we have longitudinal biomarkers because these women were screened annually. And then we have tried, of course, very different techniques. Originally, this project, the screening program was based on Bayesian change point models coupled with approximate Bayesian computation using Markov chain Monte Carlo technique. The idea was to find the point where biomarker in logarithmic scale. So it starts to, because cells are multiplied, it starts to grow exponentially in logarithmic scale linearly. So the idea was to use Bayesian network to find this change point. But it didn't detect all the women because, as I said, 15% of women do not display growth of the biomarker. Then we have applied, suggested a method of mean trends, so simply measure the growth of the biomarker and managed to license this technique after this paper, and it was bought by Appodia company. Then we have tried to play with multi-markers because this biomarker alone was not enough, and shown, for example, either ensemble learning for different biomarkers, or we have applied LSTM technique, recurrent neural networks, or simply considered, for example, here by the green, there are several models. There are points which were not detected just by a single marker. But now we are working on the application of Synolitic networks, and it will work like the following: that now we have these graphs, but we have dynamic evolution of this graph. And of course, there is a question how to analyze dynamically evolving graphs. At the moment, we hope that we want to compare two techniques. It's ongoing work. I have just preliminary results. We apply LSTM. So first, we apply Synolitic embedding. So we construct a graph in time. Then for this graph is a matrix, right? For if it's just a usual graph. Then for evolution, to consider evolution of these graphs, we consider LSTM technique, and then finally we apply graph neural network, and we want to use not only convolutional graph neural network, but also mostly complicated MPNN, so message passing neural network, when you have attention on neighboring nodes or edge, or GATS, so graph attention neural networks, and in this way characterize, and already ROC curves looks much better. Or we want to compare it with another technique, which is a methodology of principal graphs, actually developed by Zinoviev and Alexander Gorbany. The idea is the following: this is a kind of nonlinear multidimensional nonlinear generalization of principal component algorithm. The idea is if you have multidimensional space, and for example here healthy, and then these points develop to the unhealthy state, that you can compute a trajectory. So it's a kind of manifold learning. So you can compare a graph, so line, trajectory, so to say the path from being healthy to being unhealthy. And then along this path, you can compute the pseudo time for you towards, for the patient towards the disease. Why it's so interesting in combination of Synolitic graphs that now we can apply this pseudo-time algorithms in the space of the Synolitic graph, because we can first embed multidimensional point into the even more dimensional space of the Synolitic graph, and in this space compute the principal graph, the path from being unhealthy to from being healthy to being unhealthy. And then for each new patient, of course, you can do the same embedding and see the actual pseudo-time of the development on this path from being healthy to being unhealthy. So we want to compare what will be better if we simply apply artificial intelligence in the straightforward way, LSTM plus graph neural network, or we will apply first a kind of dimensionality reduction technique, but only after dimension will be increased using the Synolitic approach. Yeah, so now I'm speaking 1 hour, right?
And 10 minutes for questions.
10 minutes for questions. So, yeah, so but altogether it should be till 2:15, right? So I have 30 minutes for talk and questions. Super. Okay, I will finish one. Yeah, and now we want to compare this technique. So this is one challenge. Another challenge, another challenge is that Daniel Vlasenko suggested to use the Synolitic representation for analysis of fMRI images. So practically, as the age, we set to first we use map fMRI signals onto atlas, parcellation atlas of ROI, so regions of interest. There are, if I'm not wrong, 379 regions of the brain. So for each of them, the average of these regions of interest, and then between these signals, we compute the kind of the ensemble graph. Daniel does like the word Synolitic, he prefers ensemble graph. Doesn't matter. Where in contrast to the correlation graph, we measure how much these two signals altogether classify the state of the brain. For example, probability to belong to the class 2 minus probability to belong to the class 1. And then fMRI image, for example, in first our study, we applied for the experiment where a patient, it's publicly available data, a patient has seen some image, for example, the bus, and the second state, he was just imagining that he is seeing the bus. And the idea was to classify between these two states. So fMRI can be transformed into the Synolitic graph. This Synolitic graph can be analyzed with convolutional graph neural network, and accuracy actually was quite perfect. Now we are working to apply this technique for cognitive brain state classification to human cognitome project Atlas data, where you have very different states. For example, if somebody is gambling and winning versus not winning, and apply this layer of low-dimensional classifiers, construct the graph, and then apply some convolutional graph neural networks. This is done by Vadim Ushakov. I have included him in the chat about the project, so he can answer all the technical questions about the code, and this also works quite well. But one should understand that this Synolitic representation, it's some special tool, and in fact, if some marker is perfectly classifying two states, probably all other information will be just the noise for this marker. So one should probably exclude, if you want to get additional classification, one should exclude the best marker from the consideration and then do the Synolitic representation. Because by this Synolitic representation, you analyze actually 10 minutes more should be, no? Because I have still 25 minutes.
Yes.
No?
Okay.
It's till 2:15? Yeah, but I will be not so. Then we use this for active surveillance of prostate cancer, and here the problem is that patients with prostate cancer, for some of them it's not so good to apply the treatment or even the biopsy, because one can split them into the progressors and not progressors. If it's not progressing prostate cancer, it's better to put this patient under active surveillance and simply wait until it will be progressive and do nothing. And survival rate will be actually higher, because if one removes the prostate, actually life conditions will be very bad. By the way, in Russia still this approach is not used. I was speaking with hospitals, and doctors tell me, so for them it's more profitable simply to cut the prostate, because if they will, and then cancer will be treated, because if they do not do this, a patient will go to another doctor and another doctor will do this. So this is not used in Russia, despite the fact that actually life conditions after these operations are not so good. So you should go to the toilet constantly, in principle. There are a lot of problems. The idea is that we analyze here MRI images, and we started actually with radiomic parameters of these MRI images, developed a baseline model, and for this then delta radiomic model, this was actually used with paranclitic networks and have shown quite good results. And we have in delta radiomics. Delta radiomics means you analyze simply 2 points in time. Then we have analyzed actually time radiomic images when you have evolution in time. And at the moment, very new paper, then we applied LSTM. It's not so interesting, then it was even better. And just a very new paper, it was just published 10th July, so several days ago. So Nikita Sushintsev, who is actually the driver of the study in Cambridge, he has found that MRI with patients with cribriform can display especially aggressive cancer. So now it is important to predict an MRI, this cribriform, which are actually can be visualized in microscopic histopathology, but cannot be visualized in MRI. But probably with artificial intelligence, they can be analyzed. For this, I hope to couple Synolitic approach with GANs. And again, as I have already discussed with some students, the idea, because in GAN, in generative adversarial network, we have two blocks for the generation of the image and for the discrimination. I suggest that we can generate in the space of the image, but discriminate in the space of the Synolitic graph. And simply, I hope that this will be better simulation of image evolution, but again, who knows. Finally, we are now involved in the CanDetect study, so this analysis of electronic health records, 16 million people, and we were interested in dangerous gastro cancers. And here we want, so here we have just started work. We applied BEHRT, which is a BERT adaptation for the analysis of electronic health records. It shows quite good results, but I hope very much to use as well graph RAGs and dynamic knowledge graphs. So I'm showing this just if you are interested in this collaboration, because also with Samara University, we are just now prepared a paper about the same problem of analysis of electronic health for multiple sclerosis progression. And here, at the moment, this was based just on the BERT-based model, but again, I hope I'm highly interested to use knowledge graph or probably ARI graphs for this problem. Okay, actually let me finish. So let me thank my co-authors a lot, especially by Red young people who did all the calculations. And actually, thank you very much for your attention. Questions?
Okay, thank you very much for your very interesting presentation. Could you please tell that thing? So as far as understood, much of applications of Synolitic Graph framework was on dense data, dense small data sets. But,
you mean not sparse data, without missing values?
Yes, so did you try to apply your framework to large sparse data sets like mentioned electronic health records, and what are results, how they perform compared to
yeah, that's a very interesting question. Just let me answer this and then we will continue.
Yeah,
we have not yet played with sparse data, but in fact this graph is a set of independent classifiers. So I assume that if some data are sparse, first of all, you can still compute a Synolitic graph, no problem. In contrast to other algorithms, for example, you can't apply KNN, but no problem with quadratic Synolitic graph. Of course, the question how the sparsity will affect the topological characteristics of this graph, but I hope that if you analyze this graph with graph neural network will be able to fight against sparsity. I don't know. Yeah, that's extremely interesting question and important because in clinical practice usually there are a lot of missing data. Of course, you can use in different kinds of imputation, but can we trust imputation? It's always a question. So my suggestion is simply, since there is no problem to construct a Synolitic graph, simply to check. And by the way, this is one more question for the research project if somebody will do this. If you sparse the data and simply exclude some points, how much classification results will change? Very important, very interesting.
I have a question. So for this, it's important first to convert the data into embeddings, right? To apply the method.
You mean embedding?
Right.
Yes, in graph space, yeah. So we embed vector to matrix.
Right, but to train the embedding model, you need data. So do you understand correctly that kind of limitations of this approach is if we have some specific data set, for this data set we need to have some pre-trained embedding models.
Some what?
Pre-trained embedding models. Say we are working with MRI signals or
we just need a training set of data which are labeled. So it's kind of supervised learning because you have to construct this layer of low dimensional classifiers. Yeah.
Okay. And we don't say, if we are talking about with time series data like EEG data, we don't need any RNN or something which produce embeddings.
Which produce what?
Embeddings.
For time series, probably you need initially some embedding.
Some pre-trained model.
Like encoder, yeah.
Yes, so we need some encoding.
Yes. Because at the moment I do not know. Of course, you can apply Synolitic graph at any time point, but then it will be the same frequency in the change of Synolitic graphs. So in fact, you don't need this, and maybe, but will it work or not, I do not know yet. We have not yet enough experience with high frequency.
And what's about fMRI data, because it's a sequence of three-dimensional images?
Yeah, we didn't need any encoder, no.
In this case, we don't. Okay.
No, yeah, thank you, yeah.
Hello, thank you for your lecture. My name is Romanov from Protect Digital Solutions and ARI. It's just a continuation actually of the Evgeny's question, because right now I'm working with electrical data in industry, and I think that the correlation analysis and graph neural networks can help me to solve my tasks of detection of anomalies in the equipment. And my question is, the main problem in industry, first of all, it's data, but since there is simple classifiers, it's quite easy to neglect this problem. Can we use it somehow for domain adaptation? Because there is a problem then the signals has different distributions because they change the equipment, we change...
And again, which signals, EEG or what kind
electrical, it's actually current, voltage and vibration.
Vibrations.
Yeah, it's actually pretty high frequency, around 20 kilohertz, etc.
Does it have oscillatory nature?
Yes, it's sinusoids because it's
Then
currents,
I
it's
would
periodic data.
Yeah, then I would suggest first to exclude these main oscillatory attractors and consider deviation in around these attractors. For this deviation, then apply Synolitic approach. In fact, there is another idea. So these methods of principal graphs exist now for not oscillating data. I was thinking probably one can generalize it for oscillating data. Why not to identify principal attractors? So in fact, you can, no, it's like any kind of spectral, you can...
Yes, it's one of the techniques in field.
You can expand your signal into different frequencies, or probably you can expand your signal into a different set of different attractors, principal attractors. So I would suggest first to do this pre-processing, because in my experience of working with EEG, if it's highly oscillatory, if you directly apply any encoder or Synolitic approach, these oscillations will kill all the information.
Yeah, or maybe use just the statistical features, which, for example, integral, linear integral, etc.
If you are sure that it's stationary, maybe you can consider always dynamical spectrum instead of the signal. So
it likes
embedded in the spectral space, and then play
wavelet transformation.
Or wavelet transformation. No, start
time.
with simple spectrum without wavelets, then additionally you can apply wavelets. But if you have these dynamic attractions, maybe attractors, maybe you should expand it into the set of principal attractors. So I would be very much interested to play with it, in fact. So very happy to discuss. Please don't hesitate to write me or...
Okay, thank you very much.
One more question about the meaning of weights in Synolitic neural network.
Meaning of what?
Meaning of weights.
Weights, yeah.
As I understood correctly, the higher the weight, the more important features if you want to classify the patients.
Yes, yes.
And have you tried some weights with other meaning, for example, cross-correlation of two features or mutual information? Do they give the same results?
For cross-correlation, you need time series. So here, actually, I was mostly...
Or maybe mutual information or some other...
For mutual information, yeah, you need just distributions, but...
they are not directly applicable to this.
We use, for example, mutual information when we calculate integrated information, so level of consciousness of some black box. Whether mutual information is so good for classification, maybe yes, maybe not. Important point is that this kernel, so how you construct this classifier, it doesn't matter, it's quite universal. You can substitute here a classifier based on mutual information. Why not? Just two features, two distributions.
Okay, thank you.
Yeah. In this case, it will be still Synolitic approach, which is an ensemble of low-dimensional classifiers represented as a graph.